Today we will continue the genetic engineering. In last few classes, we discussed about the first two major steps of genetic engineering. In the first step of genetic engineering, we have to construct a recombinant DNA. Recombinant DNA means the DNA that created by joining two different DNA from two different sources. For example, one human gene and another bacterial DNA ligated, so that is a recombinant DNA. Now question arises, who first constructed recombinant DNA? This is your answer. Herbert Boyer and Stanley Cohen. In year 1972, they first constructed recombinant DNA. One more question. So how they constructed recombinant DNA? For construction of recombinant DNA, two different DNA sources required. Here, they selected one bacterium, Salmonella typhimurium. Suppose this is the Salmonella, and from the plasmid of Salmonella, they isolated one antibiotic resistant gene. Suppose this is that antibiotic resistant gene. And again they isolated one plasmid DNA from E. coli. By means of restriction endonuclease, they caught the both DNA. So what they got? They got sticky ends in both DNA. So by ligation, with the enzyme DNA ligase, ultimately they successfully constructed recombinant DNA. So what are the two DNA sources for construction of rDNA by Herbert Boyer and Cohen? Now, antibiotic resistant gene isolated from plasmid, ligated to the plasmid of E. coli, and again this recombinant DNA was introduced inside the E. coli, because Salmonella typhimurium and E. coli, they are very closely related bacteria. Means the plasmid can replicate in the cytoplasm. So this cytoplasm will provide the environment for its replication. So this part is over now. Now question arises, who is the father of genetic engineering? Answer is Paul Walsh. So Paul Walsh, in year 1972, in the same year, they started engineering of the gene. What they did? This is SV40 virus. This is a cancer causing virus. Simian virus 40, SV40. Paul Walsh isolated the DNA from the SV40 virus and introduced that DNA into another bacteriophage. Bacteriophage, this can attack bacteria cell. This is a lambda bacteriophage. SV40 cannot attack bacteria because it can attack only mammals. In first year, you know, viruses are very specific for their host. Human virus cannot attack plant cell. Like this, SV40 virus, this can attack only animals, mammals, so that never attack bacteria cell. That's why Paul Walsh isolated the DNA and introduced into the bacteriophage, and this bacteriophage was allowed to attack, so that SV40 gene ultimately introduced into a bacterial cell. So this is nothing but engineering of the gene. एक से निकाला दूसरे में एंटर किया. This is engineering. So in 1972, Paul Birch performed this demonstration, and in the same year, Cohen and Boyer artificially constructed recombinant DNA. Very important. Now come to here. So, what is the purpose of genetic engineering? The purpose of genetic engineering is to obtain gene products in an industrial scale. So, in that sense, I told you code making is also a traditional biotechnology. That is also a product due to the activity of microorganism. So after construction of recombinant DNA, the DNA that contains gene, we have to introduce the rDNA inside a cell. Suppose this is our recombinant DNA. Again I am drawing here. This recombinant DNA can enter into any type of cell. Suppose these are the bacterial cell. Last class I told you what are the various mechanism of gene transfer. The gene transfer mechanism depend upon the host cell. If the host cell bacteria, which type of gene transfer we can proceed? That is a hit-shock method. If the host cell is a plant cell, we can do gene-gun or biologistic method of gene transfer. Otherwise, agrobacterium mediated gene transfer. Clear or not? If these are animal cell, how we can transfer gene into the animal cell? Either we can do micro-injection, or we can do microporation, or we can introduce the gene by means of using disarmed retroviral pathogens. Retrovirus, so disarmed retrovirus containing harmful gene, and we will cut and we will introduce so that it will be insertionally inactivated. In one word, disarmed. So by using disarmed pathogen, we can introduce. Suppose these are bacterial cells. By hit-shock treatment, we introduce the recombinant DNA, and interestingly, we don't know which cell is having the recombinant cell. So the cell having recombinant DNA is called a transformant. We can say it recombinant, and those are not getting any rDNA, they are called non-transformant. So this is our transformant, and these are non-transformants. This is our non-transformants. Transformation, so these are the transformed cell or transformants. Now, today we will discuss about the gene cloning. Cloning means what? To create multiple copies of identical molecule. Here our molecule is DNA. So making multiple copies of identical DNA is cloning. Gene is a DNA segment. The DNA may contain a gene. That is a DNA segment. So gene cloning, otherwise called amplification. We know how outside cell we can do amplification. That is PCR technique, polymerase chain reaction. So by PCR we cannot obtain gene product. Our main purpose is to obtain gene product. That's why we have to introduce that gene inside a cell. Then only in the cellular environment, the cell can, the DNA can express by transcription translation. Then only we will obtain gene product. So gene cloning, it is the increase or multiplication of DNA number. By gene cloning alone, we cannot obtain gene product. That's why we have to introduce the recombinant DNA by gene into a cell and culture this cell in a culture medium. Then only we can obtain gene product. So the stainless big vessels in which we culture these recombinant cells, that is called as fermenter or bioreactor. Before that, I want to discuss with you how we can separate transferment from non-transferment. If I will give food material, the non-transferment, they will enjoy the food material. In return, they will give nothing. So, by means of chemical engineering, I will prepare such type of culture medium so that transferments will survive and non-transferment will die. Then indefinitely I will start culturing. जितना culture करो, transferment का volume बढ़ेगा, and my product, I will get my product also. So here we will discuss how we can separate the transferments from the non-transferments. All these procedure, this process is the transformation. All these things I have done inside a test tube. That means it is in vitro method. Suppose all these cells is present in this test tube. So this test tube contains transferment cell, non-transferment cell. So what I will do, I will transfer this into a culture medium. This is a glassware, that is a petri dish. In botany, zoology, microbiology laboratory, small glassware there, that is called petri dish. In that petri dish, I introduce a culture medium. I told you, agarose gel is the culture medium for laboratory work. So agarose gel is neutral. I added some food material, artificial nutrients to the culture medium. That is the chemical engineering. So in the surface view, see here, this is the surface view of the petri dish. Petri dish को ऊपर देखिए, surface view. So I pour the entire content of the test tube here. When the agarose gel is liquid, I added the culture medium to the petri dish. After few minutes, agarose solidified, basigola. And after 1 or 2 days, we can notice the colony. Suppose I selected 3 bacteria. Suppose 5 bacteria. In the culture medium, 5 bacteria. Out of these 5 bacteria, any one bacteria is having recombinant DNA, that I don't know. So, initially a single bacterium cannot be seen in naked eye. ଯେତେବେଳେ ମୁଁ ତାକୁ କଲଚର ପାଇଁ ଛାଡ଼ିଦେଲି, They started doing binary fission, they multiplied, they increased their number by binary fission. Culture medium रे गोटे बैक्टीरिया रा कॉलोनी तने गोटे बैक्टीरिया सेल डिवीजन करी गोटे कॉलोनी हिची तने जेतेटा कॉलोनी मते देखा जाउची खाली आखिरे सागदाना वाला देखा I can see them. तने these are the bacterial colony. तने any one colony contain recombinant DNA. Suppose a colony का recombinant DNA जानिया केमती तले I have to apply some technique. For that I told you the vector DNA must contain 2 antibiotic resistant genes. These are marker genes. This recombinant DNA, suppose it is the PBR322. P stands for plasmid, B stands for Bolivar, R stands for Rodriguez. These two scientists, Bolivar and Rodriguez, artificially constructed the plasmid and given the laboratory number 322. तले ए रिकॉम्बिनेंट डीएनए रो दीटा एंटीबायोटिक रेजिस्टेंट जीन थिला गोटे ओरी साइड थिला तले गोटे एंटीबायोटिक रेजिस्टेंट रो जीन रो नाम होला एमआर तने दिस जीन व्हेन प्रोड्यूस प्रोटीन इट विल प्रोटेक्ट द बैक्टीरिया फ्रॉम एम्पिसिलिन एंटीबायोटिक तले दिस जीन इज सपोज टेट्रासाइक्लिन रेजिस्टेंट जीन तले जो बैक्टीरिया भीतर ए टेट्रासाइक्लिन रेजिस्टेंट जीन रहइबो से बैक्टीरिया को एंटीबायोटिक टेट्रासाइक्लिन देले भी से बैक्टीरिया मरियो एटा तो कांसेप्ट तले as per your NCERT book diagram given in page number 199 you can see the 2 cloning sites एठी गोटे cloning site अछि एठी गोटे क्लोनिंग साइट अछि दीटा क्लोनिंग साइट अछि माने रेस्ट्रिक्शन साइट जोटी रेस्ट्रिक्शन एंजाइम कटिबो one is PSTI another is PVUI similarly tetracycline also contain 2 restriction site माने दीटा पैलिंड्रोमिक साइट अछि one is BAMHI SARI now out of these 2 antibiotic resistant gene genetic engineer can cut any one gene cutting माने ए restriction site रे जो एंजाइम टा काटिबो ताको यूज करिवे suppose we have cut open the tetracycline resistant gene तामाने our recombinant DNA contain ampicillin resistant gene intact कारण my insert is in the tetracycline gene तो tetracycline gene रे मो DNA का insert करछि बोली टेट्रासाइक्लिन जीन टा इनएक्टिव हो जाईछि तले याको इंग्लिश रे कोन कहबे इंसर्शनल इनएक्टिवेशन इंग्लिश रे कहूची इन द क्लोनिंग वेक्टर PBR322 2 marker genes are there during construction of recombinant DNA any one marker gene will cut open and the gene of interest will be introduced and ligated by ligase so that the gene become inactivated the gene that is having an insertion will be inactivated and another gene is remain functional so imagine these are the colonies any one colony is having such type of recombinant DNA where tetracycline tetracycline resistant gene is inactivated. Now we have to proceed for two plating technique. In one, this is my original culture plate. I take one wooden block, जमती स्टाम्प मारिया पे स्टाम्प मारा जाए सेंती गोटे वुडन ब्लॉक नेली ता ऊपर वेलवेट कपड़ा दे देली बा नायलॉन दे देली ताको या ऊपर एक स्टाम्पिंग करी प्रेशर दे तने सम बैक्टीरिया सैंपल विल स्टिक और फांका कल्चर पेट्रिटिस सरफेस व्यू देखली तो एटा ओरिजिनल कल्चर wooden block with velvet एठानी stamping होती है ताने intentionally a transfer of bacteria पहले this is the impressions, this is my original culture, these are the culture created thereafter. So I will start adding antibiotics now. In one culture medium, I will apply ampicillin antibiotic. मु जीन टाको स्मॉल लेटर लिखी एडी कैपिटल लेटर रे स्टार्ट करी एंटीबायोटिक्स कर अनदर कल्चर मीडियम रे आई विल ऐड टेट्रासाइक्लिन. देखिए after addition of antibiotic some of the colonies disappear in both culture medium. तले ampicillin resistant जो थे अच्छी टेट्रासाइक्लिन इनएक्टिव एटी सपोज तीनटा कॉलोनी रो एटा सपोज एम्पिसिलिन रेजिस्टेंट जीन एथरे अछि आउ अन्य गुडा करे नाही तने अदर कॉलोनी विल डाई मो डॉटेड कर दूची खाली फेंट लाइन्स ताने मरगे एटा खाली बंची गला तले एम्पिसिलिन रेजिस्टेंट जीन ता इन्फ्लुएंस रे recombinant colony ता बंची रहला. That means what? The recombinant DNA is having a functional ampicillin resistant gene. बाहर एंटीबायोटिक देले मध्य एई कॉलोनी को किछि प्रभाव पड़ो कारण ताका साइटोप्लाज्म रे एम्पिसिलिन रो रक्षा करिया पे जीन ता अछि जीन प्रोडक्ट तयारी करिवो बंची देला. अदर कॉलोनीज डाइड अन्य कॉलोनी मरिगला कहीं कारण they are non transformed, they are having no DNA. PBR322 वो तो बोलते नहीं PBR322 वो बहुत गुड़े नहीं. Listen, this is a very difficult portion. Some of the PBR322 don't have any insult. भले कि सुनो किछि रिकॉम्बिनेंट डीएनए इंसर्ट हेनी जीन तामाने दीटा जीन फंक्शनल अछि ताने सही कंडीशन रे दीटा कॉलोनी बंची एम्पिसिलिन पकेले भी बैक्टीरिया मरियोनी टेट्रासाइक्लिन पकेले भी मरियोनी ताले से भलिया कॉलोनी जोटा दीटा कल्चर मीडियम रे बंचुची एई गुडा को भी हमर दरकार नै बिकॉज़ दे आर नॉट ट्रांसफॉर्मेड, दे डोंट हैव एनी इंसर्ट जो थरे जीन नाही से थोड़ी न रिकॉम्बिनेंट हला तले ए भले कॉलोनी को भी हम रिजेक्ट कर दबा ताको ओरिजिनल संगे कंपेयर करिया को कॉलोनीटा गायब हो जाउची. नाउ यू सी इन टेट्रासाइक्लिन आवर रिकॉम्बिनेंट डीएनए contain inactivated tetracycline. Tale recombinant colony that is having inactivated tetracycline inactivated ampicillin is functional. Then this colony will survive. Others will also die. Faint line here. The recombinant, the transformed bacteria that will survive in ampicillin resistant medium but cannot survive in tetracycline resistant. ताने मो ओरिजिनली ए दिटा को सहित कंपेयर करी देखिदेली जो कॉलोनीटा एम्पिसिलिन रे बंचुची किंतु टेट्रासाइक्लिन रे मरि जाउची तामाने एटा मोर रिकॉम्बिनेंट कॉलोनी ताने वर्तमान ओरिजिनल कॉलोनी दिटा टारगेट कर देली वर्तमान ताको बात देखिए के बळे काटी की अगर जेल को ताको ट्रांसफर करे वर्तमान याको कल्चर करे याको खोया को पिया को दे न्यूट्रिएंट दे let other colony die मरने दो उन लोग तो एवं सिलेक्शन करेंगे now I will transfer it to a fermenter. So what is a fermenter? Fermenter is a also known as a bioreactor. It is a stainless vessel. That stainless vessel provide optimum condition for the culturing of this colony. এই colony रो बैक्टीरिया माने कोबळे फूड मटेरियल दरकार करती बढ़िया पे केते टेंपरेचर दरकार केते pH दरकार the fermenter or the bioreactor will provide all suitable conditions. In your book you can see this is the stirred tank bioreactor. This is the two version. Most commonly used bioreactor is the stirred tank bioreactor. This is the simple stirred tank bioreactor. This is spaced stirred tank bioreactor. In simple, you can see these are the blades. When the blade will rotate, it will mix the recombinant cell with the culture medium, so that recombinant cell will enjoy oxygen, they will take food material, they will do binary fission, they will increase their number, and simultaneously their cytoplasm will also filled with gene product. Their number increase means cloning, along with cloning gene expression also taking place. Here it is called spaced stirred tank. Spaced means what? This apparatus, this rod-like structure, releasing oxygen bubble, that is called spazzer. So spazzer means release oxygen bubble. That's why spaced stirred tank bioreactor. So many apparatus are there intermittently. टिके टाइम परे घंटा परे मु तार pH मेजर कर दउथिबी ऊपर फोम ब्रेकर अछि किछि जिंस को स्टीयरिंग कले फोम फॉर्मेशन हो आई कैन ब्रेक दैट फोम by using the foam breaker. ता माने दुई रकम रो कल्चर हला कंटीन्यूअस कल्चर एंड बैच कल्चर. कंटीन्यूअस कल्चर मींस सपोज एटी प्रोडक्ट आगेला हार्वेस्टिंग कर दे ता माने इनिशियल वॉल्यूम सपोज 100 लीटर नहीं थी मो 50 लीटर डिकैंट कर दे बाहर कर दे मोर प्रोडक्ट हो जाई छी फिनिश ताने आउ फ्रेश रॉ मटेरियल भर्ती कर देली. सपोज मु मीडियम रे अल्कोहल बनौछि ताले व्हाट आई हैव इंट्रोड्यूस्ड ईस्ट सेल जेनेटिकली इंजीनियर्ड ईस्ट सेल. ताले ईस्ट सेल कल्चर मीडियम रे 13% अल्कोहल प्रोड्यूस करिवो तापरे कल्चर मीडियम का अल्कोहल एते ही जीवो जे दैट मीडियम विल बिकम पॉइजनस फॉर द ईस्ट सेल. ताले ईस्ट सेल भलिया रिकॉम्बिनेंट सेल को मु बंचिया पई मीडियम टाको बाहर कर देबे पुणी फ्रेश ग्रेप जूस भर्ती कर दे ताने दैट इज ए बैच कल्चर. रॉ मटेरियल एडिशन चलतिबो हार्वेस्टिंग भी चलतिबो प्रोडक्ट हार्वेस्टिंग. बैच कल्चर माने इनिशियली 100 लीटर नेली पूरा आफ्टर फर्मेंटेशन इज ओवर मो 100 लीटर पूरा बाहर कर देली साफ सुथरा करके पुणी बैच पर बैच कल्चर हो नले से चलथिबो कंटीन्यूअस चलथिबो टू टाइप ऑफ कल्चर तापर एथर डाउनस्ट्रीम प्रोसेसिंग फर्मेंटर बाहर करी आनली तले दैट कंटेन टू थिंग्स स्पेंट न्यूट्रिएंट्स मीडियम कल्चर सेल से कल्चर सेल को आइसोलेट करके ताको ब्रेक डाउन कले ता साइटोप्लाज्म रो प्रोटीन पे दैट प्रोसेस इज कॉल्ड डाउनस्ट्रीमिंग दैट इज सेपरेशन एंड प्यूरिफिकेशन आफ्टर दैट प्रोसेस what we will do some quality control testing whether that product is fit for marketing market ro complaint asile puni arrest hijibo tale quality control testing quality control करके found okay गोटे बैच नंबर दे गोटे सील मार दे मैन्युफैक्चर्ड डेट एक्सपायरी डेट बैच नंबर एमआरपी तले इट इज रेडी फॉर मार्केटिंग तले here we finished the first chapter of biotechnology, that is principle and processes of biotechnology. Now we start a fresh new chapter, that is biotechnology and its application. In application portion, biotechnology is very helpful for therapeutics. Therapeutics means therapy. Therapy means treatment of disease. So biotechnology can be applicable for disease treatment, for diagnostics means for diagnosis of disease. Biotechnology can be applied for disease diagnosis. Biotechnology can be used for cultivation of GMO plants, genetically modified plants, GM plants. GMO means genetically modified organisms. In agriculture, GM plants is very valuable. And by means of biotechnology, we can do bioremediation. Bioremediation means application of genetically engineered cell or genetically engineered microorganism to remove pollutants. Tale we can reduce the environmental pollution by using genetically engineered cell or transformed cell. Bioremediation. Another thing, waste production. Waste treatment, waste treatment. Then energy production like this. So first thing we will discuss role of biotechnology in agriculture. Role of biotechnology in agriculture. All of you know, in one class I told you the purpose of agriculture is food production. Again, increased food production. So traditional cultivation giving us food supply, it is not sufficient. Then organic farming, that is also increasing food but not destroying our environment. Third type of agriculture is genetically engineered crop based agriculture. That means in that agriculture we will use genetically engineered plants or genetically modified plants. That means, what do you mean by genetically modified organism? That can be three types: genetically modified microorganisms, genetically modified plants, or that can be a genetically modified animal. So all are together known as genetically modified organism. So genetically modified organism is the organism having alien gene and expressing the alien gene. That is the important definition. So what is a genetically modified organism? The organism that possess possess and express alien gene, means foreign gene. GM animal रो गोटे एग्जांपल सपोज दोची ट्रांसजेनिक माउस. ट्रांसजेनिक माउस बा माइस. ट्रांसजेनिक पिग. Transgenic cow kanala Roji. Roji is not a beauty parlor. It is known as transgenic cow. एटा तुम सिलेबस रे अछि. GM plants रे so many examples are there. One example as per your book is BT crop. This is important for your exam short note. I have written BT crops plural. BT rice, BT corn, BT brinjal, BT wheat, like this. So many crops. These plants interestingly contain gene from a bacteria, Bacillus thuringiensis. That's why BT crop. That means what? Any GM organism possesses a foreign gene, and that foreign gene is getting expressed also. Plant cell ଭିତରେ bacteria gene କେବେ ରହିବ ନା? Human ତାକୁ artificially introduce କରିଛି. ତାହେଲେ GM organisms possess and express alien gene. Transgenic mouse. ताने mouse ଭିତରେ केबे human gene ରହିବ? Human artificially introduced the human gene into mouse. Transgenic mouse. Human artificially introduced milk protein secreting gene into the cow. That's why this cow milk contain human milk protein, Roji. Then GM microorganisms. Genetically modified microorganisms. These microorganisms contain gene, suppose insulin gene. After culturing these microorganisms, we can obtain human insulin. Anand Mohan Chakravarti, one Indian born American scientist. Anand Mohan Chakravarti. Anand Mohan Chakravarti. He is an Indian born American scientist. Interestingly, he constructed one microorganism, bacteria, that is having plasmid from different bacterial sources, and that bacteria is named as a superbug. Superbug. Bug mane poko. So superbug. This is a genetically engineered bacteria. So what is the purpose of constructing this bacteria? All of you know oil spill. ସମୁଦ୍ର ରେ ବଡ଼ ବଡ଼ ଜାହାଜରେ ତେଲ ଆସୁଥିବ, जदि कौन सी पर्पज रे हेटा क्रैक करिगला ताले तेल गुड़ाक समुद्र रे वाटर पॉल्यूशन क्रिएट करे। आफ्टर फ्यू डेज एक्वाटिक फना माछ गुड़ाक मरिकी भासिबे, सील तिमी सब मरिकी भासिबे। ताले हाउ वी कैन मैनेज दैट इकोसिस्टम। आनंद मोहन चक्रवर्ती सुपर्ब को आम सांगे सांगे बिंची देले पानी रे चून पकेला बेले ताले बैक्टेरिया मानकर खाद्य कौन ना पेट्रोलियम प्रोडक्ट। सांगे सांगे से पेट्रोलियम प्रोडक्ट गुरा को एब्जॉर्ब कर नेले, डिसइंटीग्रेट कर देले, रॉ मटेरियल्स को वापस कर देले। तामाने इमीडिएटली आम एनवायरनमेंट को क्लीन कर देले ना। दिस इज कॉल्ड बायोरिमेडिएशन। क्लीनिंग द एनवायरनमेंट बाय यूजिंग genetically modified microorganisms. ताले आम साइड रे होची how biotechnology can be applied in agriculture. Under that we learn BT crops. Before that, what are the importance of GM plants? All of you have seen matchstick home light, aim, matchbox. होम लाइट मैच स्टिक लगे देले सेटा धीरे 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 पड़थो। एम टा को लगे दे टिके अनमनस्क देले तुम आंगुठी जळी जेतो। तामाने व्हाट इज द डिफरेंस बिटवीन दिस टू? ताने बायोटेक्नोलॉजी फार्म दे आर टेकिंग टेंडर नोटिस फ्रॉम मैचस्टिक फार्म, home light made by ITC company. So they are giving order supply. That biotechnology firm will receive the order, collect the gene and introduce the gene into a plant, cultivate the plant. तार किछि मास परे आनिकी देदेला आज्ञा आपण ए भलिया क्वालिटी काठ चाहुतले नियंत। ताले जीएम क्रॉप्स रो इंपोर्टेंस कोन? These are used to provide tailor-made crop plants. In coming class, I'll begin from here. This much for today.